The second macromolecule we'll consider are nucleic acids. The building block for nucleic acids is itself somewhat complex. The innermost part of a building block is actually a sugar, a pentose. And this pentose, a 5-carbon sugar, is called ribose. And ribose is what is used in RNA. In DNA, it still uses ribose, except instead of this hydroxyl, there is only a hydrogen. There is no oxygen, and hence the name deoxy, no oxygen. Okay, one of the oxygens is missing, so it's deoxyribose. And deoxyribose is what's used in DNA. Okay. So the ribose plus a base, a nitrogenous base, so a nitrogenous base is linked to the ribose of deoxyribose. And there are 4 kinds of nitrogenous bases in DNA and 4 kinds of nitrogenous bases in RNA. Both DNA and RNA have the same purines. These are adenine and guanine. Purines have 2 rings, a double ring structure, okay, with nitrogens in them. And pyrimidines all have a single ring structure. Okay. RNA has cytosine and uracil, and DNA has cytosine and thymine. So the sugar plus the base is a nucleoside. Once we start adding phosphates, we get nucleotides, and you can have between 1 and 3 phosphates. These nucleotides then are linked together in a straight chain, a linear chain, by what's called a phosphodiester bond. And what's shown in blue are these, or in the blue, inside the blue circles is a phosphodiester bond. Okay. And so we have sugar, phosphate, and this part of the molecule is what we call the sugar-phosphate backbone of either DNA or RNA. And then the sequence of bases is what we call the primary, or what we could call the primary structure, is a linear sequence of bases. For example, A, T, G, C, C, and so forth. Now, nucleic acids can also have secondary structure. You know very famously that DNA is a duplex. It takes 2 strands to form a duplex DNA molecule. And these 2 chains of DNA are held together by hydrogen bonding between the base pairs, as shown here. So the hydrogen bonding between the nitrogenous bases is what gives nucleic acids their secondary structure. So the secondary structure of DNA is simply the DNA duplex formed by complementary base pairing and hydrogen bonding between the bases. And RNA molecules, even though RNA molecules usually consist of a single RNA strand, the RNA strand can have secondary structure through pairing between bases within the molecule. So this is an internal base pairing. So these are 2 examples of RNA structure. One is a stem loop, and this more complicated structure is called a pseudoknot. Mainly, I just want you to be aware that nucleic acids do have secondary structure, and that the secondary structure is held together by hydrogen bonding between bases. Here is a handy chart for you to compile all of this information. Polysaccharides and nucleic acids are the molecules we have talked about, and I urge you to go ahead and fill in this chart to help you remember and organize information about these two classes of macromolecules. Next, we will talk about proteins.